We'll get started. So welcome to this webinar organized by the Convention of Biomedical Research, Ghana. This is continuing in our series of webinars to share cutting-edge research and also to keep abreast with current developments in bioscience. This morning, it's my pleasure to introduce to you Professor Terrick Andey. Professor Terrick Andey is a product of Prempeh College, Amanfour. He's also a graduate of KNUST, Faculty of Pharmacy. He holds a Doctor of Philosophy degree from Florida in the United States. Currently, he's an Associate Professor of Pharmaceutical Sciences. His work is in the area of drug delivery, nanotechnology, and signaling in cancer. So without further ado, ladies and gentlemen, Dr. Terrick Andey.
All right. Thank you so much, Dr. Comensa. Can you hear me?
Yeah, I can hear.
All right. Thank you so much for inviting me to speak at this webinar. As you said, my primary area of scholarship is in drug formulation and delivery. That interfaces with molecular biology, pharmacology, as well as aspects of biopharmaceutics. So I do genetic profiling and others. Starting, well, I joined this university back in 2015, and that afforded me the opportunity to work with Dr. Comensa. Hello?
Yeah, go ahead.
Right. Okay. Who's in Dr. Comensa's area of scholarship is in computational biology. He's done so much in that work. And so we kind of started working together, trying to integrate my formulation approach with his computational biology approach to try to understand targets, identify and, targets for some of the formulations that we come up with. And so for me, it's been a learning process as it has been, a scholarly interest as well. And so for today, I'm trying to join together my primary area of research and formulation with our curiosity in systems biology, systems oncology, in order to effectively target cancer. All right, so a brief outline of my talk would touch on cancer, the etiology, the historical trend, and then the pathophysiology of cancer. And then I'll briefly talk about, diagnosis and treatment and prevention of cancer. And then we'll be looking at the computational biology aspect of it. So for this, I'll essentially be concentrating on the Cancer Genome Atlas and how data that is has been collected by this consortium can be used to essentially guide our treatment of cancer. Under the systems biology, I'm specifically looking at tumor resistance, so drivers of tumor resistance. And in this regard, I would be talking about cancer stem cells, and then dynamic rewiring in cancer cells. And then finally, I'll bring it all together with the formulation aspect of it in order to improve therapy. So this is essentially a conceptual image of a tumor, a solid tumor. And as you can see, it looks very uniform in terms of the morphology with some nodules here and there. And then you have blood supply essentially coming out of the tumor. And that's very necessary to ensure that the tumor continues its growth. And I received a question about, essentially targeting this blood supply of tumor, which I'll try and then answer at the end of the presentation. All right, so briefly, a lot of people are interested in what causes cancer. Now, that is still up for debate. What has been shown is that there are risk factors associated with developing a particular kind of cancer. So there's no consensus about what exactly causes it, even though we have a lot of evidence to kind of support a causal role of some causes. But generally, it's understood that certain risk factors would predispose someone to developing cancer. And some of these risk factors may be genetics or any familial predisposition to cancer. Hormones, it could be a chemical or the environmental causes, it could be an autoimmune disorder, lifestyle, so lack of exercise or poor diet, leading to such conditions as obesity, for instance, have been linked to development of cancer. Certainly radiations are also known to cause cancer, as well as infections. And in this case, we can talk about the HPV virus, the human papilloma virus, as, for instance, one cause of cervical cancer that we know of. Now, briefly, I'm going to try and then capture the historical trend of cancer from antiquity up to the present time, which is difficult to do, but I'm trying to do that in one slide. So we know that cancer or tumors were known way back. So back in 3000 BC, the scientist Edward Smith, his papyrus essentially cataloged breast tumor surgery. So as way back as that time, there was evidence that they were very much aware of cancer, and they had surgical procedures to remove those. And between 460 and 370 BC, Hippocrates actually coined the terms carcinoma and carcinoma to describe a tumor. And then later in 2080 to 50, we had Celsus also coining the term cancer to describe a tumor. And then after that, we had Galen introducing the term oncos, from which we get our oncology, the study of neoplasia or tumor growth. And then in 1705, we became aware of the hereditary link to cancer. And then in 1761, tobacco snuff was actually linked with nasal cancer. So we had a chemical component, a chemical environmental component to cancer. And then from the 20th century up to the present time, we've seen a rapid advances in the diagnosis and the treatment of cancer. And it's in this present time that I'll be essentially, talking about the approaches that we are adopted in trying to overcome cancer. So in terms of understanding the cancer as a disease, there are different tumors present with different features that we call hallmarks. And we also are very much aware of the association of mutation, which is a genetic aspect of genetic aspect to the development of the cancer development and then the progression of cancer. And then we also are aware, and this is much more recent, of the epigenetic causes of cancer. So these are modifications of the chromosome that do not necessarily change the DNA, but they affect the function of the proteins that are finally produced, which actually carry out the biological function. So these are widely regarded as important, features of cancer as a pathology. And most of the treatment strategies that we are looking at essentially tries to target some of these hallmarks. So in 2010, in the year 2010, and then a couple of researchers came out with an essay or a review that tried to summarize what we knew about cancer until then. And then later, a year later, they came up with a revised version of the same, essentially supplementing it with new knowledge that identify the hallmarks of cancer. So what actually are the underlying framework of cancer? Now, one of those is sustaining proliferative signaling. So cancer cells or tumors have been known to have very high proliferative potential. So they keep on growing, dividing and growing. And then they also have the capability of evading growth suppressors. So the cell has a very intricate machinery that essentially has checks and balances on how that machinery works. But then cancer cells, unlike normal human cells, would have essentially found a way to evade some of these checks, and then essentially going beyond what a normal cell would. And then there are also other aspects of cancer that has to do with invasion. So you have activation of invasion and metastasis signaling. And then there's this other aspect of it, which was more recently identified, which is enabling replicative immortality. So essentially, this kind of presupposes that cancer cells do not die. They just kind of, keep growing, keep dividing. And this is the point where I'll be introducing the cancer stem cell theory later on the presentation, because it essentially dovetails into the replicative immortality theory. And then induction of angiogenesis. So as I showed in that picture in the second slide, cancer cells essentially have the capability of not just co-opting the blood supply of normal cells, as we used to think in the past, but they can actually generate their own blood vessels. So they kind of, recruit endothelial cells and then essentially start generating or building their own blood supply. So it's a very dynamic process when it comes to cancer. And then finally, resistant cell death. So as we know, all normal cells would essentially, die at some point. The cell actually has a program suicide, machinery that makes sure that cells that are compromised or are old essentially will be cleared out of the system. Now, cancer cells have found ways to actually evade this this restriction on growth and immortality, and this is one major hallmark of cancer. So, to put it together, to kind of capture all of those 6 frameworks under one framework, essentially 2 frameworks, cancer cells can be said to be essentially driven by instability in the genome, so that the full complement of the genetic makeup of cells. And when there is instability in that system, it kind of generates a diverse population of cells, and that diversity actually promotes the acquisition of new features that then makes the cancer sometimes different from its initial form, by acquiring new features and then becoming much more lethal or difficult to treat. And then second, and one very important framework is the occurrence of inflammation. So we do generally know that cancer is a genetic disease, but we also know that cancer is an inflammatory disease. And almost every cancer cell would have a very high inflammatory potential. You have high signaling when it comes to inflammatory processes, and these also promote the growth and then of the cancer and the difficulty in treating it. Now, if we take a normal cell, essentially, we know that we have the nucleus, and within the nucleus you have the the DNA within the chromatin material, and it's this DNA that would promote the transcription, right? And then essentially of genes, and then essentially the translation of proteins. So the production of the protein as an endpoint product would start in the nucleus. But when it comes to cancer cells, essentially, you could have many changes occurring in the genetic makeup of the cell. And one of those is mutation. This is a key point of most of our cancer research right now, recognizing that genetic mutation is a major driver of cancer growth and cancer resistance. And there are very many different types of mutation. So for example, we have a missense mutation, where unlike in this normal sequence of amino acids, right, you have, the production of a different amino acid based on the insertion or replacement of a nucleotide, right here. And this would result in the production of a different protein that would, essentially carry out a different function or affect some genetic, process. You could also have nonsense mutation, right? In which case, a replacement of a single nucleotide will result in a stop codon. So essentially, the production of these other amino acids would stop because there's a stop codon right there, and a protein is produced that has no functional role in the body other than to prevent the kind of production of the normal protein that is needed for physiologic function. And then you could also have an insertion mutation, where you have an insertion of a nucleotide, as I'm right here, that will result in a protein being formed, but then again, that protein will be malfunctioning because it wasn't the right protein needed to carry out that biological or physiological function. And then you could have a whole deletion. So essentially, you have a deletion of a single nucleotide that would then result in, a whole set of new amino acids being, amino acid sequences being, added, which again would result in an alteration in the function. And then you could have a frame shift. So in a frame shift, essentially, you have just a change in the whole sector of the gene, right? So the whole sequence is just taken out, and this essentially introduces again a new set of protein that is produced that will then affect the physiologic function. And then you could have a repeat expansion. So essentially, you have this amino acid essentially produced, but then you have it repeated a number of times, and that again introduces some instability. And finally, you could have a duplication where a section of the chromosome, so the gene right there, is duplicated. And again, that could affect the function of the protein. So we have all these variations being introduced by mutation that can result in very many different kinds of diseases. And these are some recognized mutations that occur in a lot of cancers. Now, to give you an example, we have essentially a deletion right here. So in this normal chromosome, we have a chromosome pair, a chromosome 9, and then 22. And then what we see here is that at this position, you have this gene, the ABL, encoding this, the ABL gene encoding a protein, and then the BCR gene, right? And what happens right here is a breakage of the ABL, right? And then it's binding to a portion of the chromosome 22, forming a fusion protein. So you have a BCR, ABL, a fusion protein, which essentially becomes like a tyrosine kinase receptor. It becomes more like a pro-growth receptor. And that promotes the production of some genes, proteins that may essentially promote cancer. And this fusion protein has actually been implicated in a lot of leukemia cases. And the new chromosome formed is called the Philadelphia chromosome. So this is a very practical example of how, let's say, a translocation of a gene from one chromosome to another results in an unintended chromosome, which will then drive a certain pathology. Now, we also said that changes that happen not during the kind of, the translation, the formation of your messenger RNA or in the DNA, but changes that happen outside of that, changes that happen on the protein itself, also promotes some aspects of, of cancer. So in this, essentially, we have a chromosome that along its, sequence, essentially, you have various epigenetic molecules, what we call these histones, and these introduce certain kind of changes. For instance, they may introduce methylation, so methyl groups in there. And cancer, for instance, some cancers have been shown to be associated with very high hypermethylation of the certain gene. So this epigenetic change, right, that happens sometimes outside of the gene, right, outside of the DNA, will result in a protein that would have certain groups attached to it, like methyl groups, like acetyl groups, and those would then result in, certain kind of cancers that would then, present with their own features. And similar, right? So right here, we try to emphasize how um, certain modification, for instance, with histones, would actually promote the expression of a certain gene and ultimately a protein, whilst other histones or other modification would then result in the repression of the same gene. So you could have, for instance, methylation resulting in, um, activation of a gene, and maybe acetylation resulting in repression of a gene. And all of these very intricate changes affect how the final protein that is produced would function. And that have been shown to have implications in cancer as well. And to kind of emphasize it further, here we have a genetic instability, all right? So we have an oncogene called KRAS, and when a certain gene was silenced, it resulted in very high mutation or gene alteration occurring, right? And that introduced, tumorigenic growth. So you had a lot of tumor growth occurring based on this mutant KRAS, right? And then when we look at the epigenetic component, for instance, you have a protein from the PDGFRA protein from this gene. This is the gene that encodes it. And this uh, protein essentially may be affected, or its expression may be affected by mutation that occurs in this gene, the IDA gene, right? Which will then promote hypermethylation. So there's a lot of methyl groups that are, introduced into the final protein. Some of that mutation is inconsequential, right? But then others may also result in, the formation, production of essentially, a high tumorigenic growth, leading to a different form of, phenotype of cancer, sometimes different from what you have with a genetic, change or mutation that happens. So you have essentially the point I want to make over here is that the development or the cellular biological machinery that underlines the growth of cancer cells is very, very dynamic and very intricate. There are so many different things to go wrong in that very dynamic, system. And that challenge is, very, it's not lost on a lot of scientists and researchers who are engaged in targeting cancer. But with this, we are still, looking for ways to be able to overcome these different instabilities and promote effective treatment of cancer. So here, as I showed you in the earlier slide, where we're introduced to the 6 hallmarks of cancer, there's a certain kind of a convergence of the genetic and then the epigenetic, and there's also this kind of understanding of the diversity of tumors. Different tumors have different properties, so no two tumors or cancers are the same. It's difficult to treat the same different tumors with the same treatment because they present with different mechanisms and factors that promote their growth. For instance, in this developmental glioblastoma, which occurs mostly in adults, right? We see that most of the oncogenic changes or the promoters of this protein are mostly genetic changes. So these are mostly genetic, mutations that occur, right? So the epigenetic mutations or alterations are those that are colored blue. And we can see here that TET, which is an immortalization signature, may have, equally epigenetic and genetic components to it, which may then, promote cancer. But mostly all of these, oncogenes, P10, EGFR, RB1, and CDKN2A, are in glioblastoma, essentially, are affected by their genetic instability or the mutation. Those are the drivers that promote this kind of tumor, which is very aggressive. If you guys, if you remember, John McCain, who was a senator in the U.S., for instance, passed away because he was diagnosed with glioblastoma, and I think within a year he was gone. And before him, his very good friend Ted Kennedy, also was diagnosed and he passed away. So there's a very aggressive kind of tumor, and you can see that most of the genetic changes or mutations that promoted are mostly mutations, right, in these oncogenes. And then here, for instance, we have astrocytoma, again, a kind of, tumor in the brain, which has kind of equally changes in the genes or genetic mutations, as well as epigenetic mutations, so changes that happen, outside of the chromosome, right? And so, for instance, you have TP53 mutation and ATRX, which are, oncogenes. And then you have the epigenetic component being hypermethylation of certain genes, in this case, the IDH1, right? That then drive this kind of tumor. And then finally, we have the ependymoma, right? And this is a kind of brain tumors that occur mainly in pediatrics, mainly in children. So right here, we see that between adults and then children, there's both a difference in the causes or the predisposing factors. In the childhood cancers, for instance, it's mainly epigenetic. It's mainly changes that happen outside of the chromosome. Whereas in the adults, it's changes that happen in the gene, right? It's more of genetic mutation. So these kind of appreciating how these different differences occur in tumors helps us to essentially figure out the best way to target them, to treat them. Unlike, in the past where all that we had was, chemotherapy and radiation and surgery, here, when we're able to identify the genetic causes, for instance, if it's a mutation of a gene or, epigenetic changes to a particular, gene or a protein, we can develop a strategy or a therapy to target that specifically. And that essentially improves the outcomes for the patient. Now, so as you can see, genes play a very vital role in our understanding of, cancer and the way to effectively treat them. In 2005, the U.S. government under President Bush, I think, was it 41, Bush 41, essentially commissioned a consortium or a project called the Cancer Genome Atlas. This was under the auspices of the National Institutes of Health in the US, but more specifically under a department called the National Cancer Institute. And the aim of this group or this program was essentially to bring together scientists and researchers and research institutions from all over the place, form a consortium, and try to determine the genetic, the full genetic, basis of different tumors. So the aim was essentially to sequence all the protein, all the tumors that they can lay hands on, and try to identify the genes that are driving these kind of tumors in order to develop, proper therapies for them. And what this project essentially ended up giving us was a whole lot of data, whole lot of genes and mutation and all that stuff. And the next challenge was essentially how to use them. Right. Now, in trying to appreciate the work of the Cancer Genome Atlas, what we have to do is to essentially, again, appreciate how different human beings are. So one person, can develop different tumors in the body. So you could have there could be a primary tumor, let's say in the lung, and then which may then metastasize to the brain or the bone marrow or, to the breast. But then these different tumors would be different from each other. They will be different from, those in other metastatic sites and different from the one in the primary site. So even within the same human biological system, different tumors would have different biology, right? And so you can imagine how different tumors from one person will be to, compared to another person. What the, TCGA wanted to do essentially and set out to do was to try and gather all the samples from different patients, essentially kind of pull them together and then somehow identify the central drivers of, let's say, certain tumors, right? So let's say mutation data and all. And then try to inform how we go about treating them. So this has been the major kind of goal of the Genome Atlas. And when it was started in 2005, the initial budget was $100 million. And over the years, up until last year, I think they've taken in more than $500 million. So it shows you how important this project has been to America and generally to a lot of scientists in this area. So to kind of, put it together, I talked about data, the amount of data that was generated. So this is what essentially, they have about 2.5 petabytes of data, and this is the equivalent number of DVDs that you'd need to put this data on. So that's a lot of data, right? So these are genetic data and mutation and all that stuff. And related, these data from 33, different tumor types, right? Including 10 rare cancers, and had at least 11,000 patients contributing sample to this, right? And these then were essentially curated the data and then presented in different forms. So you may have like more than seven different data types out there. I'll be talking about some of the research institutions who've been involved in the TCGA. And out of this data, essentially, three, groups of data or results were kind of obtained. Essentially, the data that we got from the TCGA gives us a molecular basis of cancer, right? So it gives us a very genetic basis. So all the genes and the proteins and the that are kind of driving, the cancer. And then it helps us to actually essentially subtype the cancer. Within breast cancer, for instance, there are many different subtypes. So in treating a breast cancer, in fact, it may be difficult to get curation or cure or remission if you go about using the same treatment across different patients, because you could have different subtypes presenting, right? And this data helps us to kind of appreciate these different subtypes. And then because it yielded a lot of targets or mutation, genes that were overexpressed that were identified to be oncogenes or drivers of tumors, for instance, were identified as targets, and then therapies were developed, right? And they have like about at least 20 collaborating institutions who have been part of this consortium. So essentially, once that 2.5 petabytes of data had been obtained, the next challenge was what to do with it, right? It's not just useful to have data. It's how do you interpret the data and then, make it useful. And they've tried to kind of, present the data in different forms, right? So some of them may be imaging data. Some of them may be proteomic data. So for instance, the Cancer Proteome Atlas, which is hosted by one of the institutions, MD Anderson Cancer Center, is more about the proteins, right, that are involved in these promoting cancer. We have so many other, institutions that are involved, and they have their signature, product that essentially tries to present different aspects of the data that the Genome Atlas was able to obtain. You can, essentially based on what you are looking for. If you are looking for, let's say, non-coding RNAs, go look for it. If you're looking for survival data, right, so genes that are associated with survival, for instance, SelfNet by MD Anderson Cancer Center essentially is a place to find it. So you have all of these, going on. Then you have Integrative Genomics Viewer, where it tries to, shows the link between, genes and how one gene can affect the other. So these are different aspects. So for instance, one of the portals, one of the programs is the CBio portal, right? And the CBio portal essentially is, yeah, it's a product of the Memorial Sloan Kettering Cancer Center. And when you go to their webpage, this essentially is, what greets you. So you have about almost 11,000 samples out there, including mutation data and copy number alteration data. This essentially, captures changes in the chromosome. So for instance, there's, more of the copies of the chromosome produced. And under this, you have like many cancer types captured, right? And then you have mutation, the different mutation that are there. And then you look at, the specific genes that drive these mutations. So you see that TP53 up right there, like accounting for almost 42% of mutations in these. You have some fusion proteins, for instance, or genes that promote certain growth in the cancer, and a whole lot of data. So this is a form of data presentation that the CBio portal, for instance, gives you. And so when you're interested in, for instance, one specific gene, you can check on that gene or that protein, and then that will take you to its break it down for you and show you, all the different cancer subtypes that are affected, and other proteins or other genes that it interacts with, and how that promotes the cancer. All right, so let me pause right here and ask. Okay, Dr. Akomensa, how much time do I have left? Okay. I guess I'll keep on going. Before I continue, does anyone have any question for me? All right. Okay. All right. There'll be a section at the end, and you can ask any question. All right. So right here, I mean, this is essentially a kind of a conceptual image of various interactions that are occurring, between different genes or different proteins involved in certain kind of cancers. And then you can see, the connecting lines that connect the dots, right? So you can see a node here, and then, it springs out to other nodes, maybe a gene or a protein. And this complexity is very real when it comes to cancer, right? So you can have thousands and thousands of genes that may be in the cancer after it's been sequenced, that you have all these genes, and they're able to show that, oh, there's mutation in this gene. There's mutation in this gene, or there's increased expression of this gene. And what it shows is that this gene doesn't necessarily live in isolation, right? it is connected to other genes or other proteins or what have you. And so these connections are important for us in trying, in our, attempt to try and treat these cancers or target these particular molecules. And that's where what's important. It's not just recognizing that there's a mutation in this gene or there's increased expression of this gene. It's about beyond this gene, what are the factors? What are other components of its activity? Or what are other players that it's connected to? How are they affected, right? So that we can have a much more holistic approach to treat it. All right. So let's essentially I just kind of introduce you to kind of the systems, it's system biology, targeting of cancer. What I want to emphasize on is how these different molecules, right, promote the resistance that are associated with cancer. So the challenge with cancer is that it's not just good to have remission occurring. It's important to have cure. And as of now, there's really no cure for cancer, because for every cancer that goes into remission, there's always going to be a recurrence. Every cancer. So that has been a major challenge for treating cancer and making sure that it doesn't recur. So as you can tell, my interest is really in targeting this resistant phenotype of cancer beyond just clearing the tumor mass. How can we make sure that it doesn't come back? Because usually when it does come back, it's really, really aggressive, much more aggressive than it was, in the primary step. So in trying to understand that there are very many different components of resistance, but I want to talk about one aspect of it, which is what we call the cancer stem cells. Now, this is a new paradigm that was introduced, within the past 10 years, which kind of recognized that very similar to normal, stem cells that occur in the body. For instance, when you take the development of blood cells, for instance, you have an initial, stem cell that self-renews, so it's kind of, can regenerate itself. That matures into a multipotent progenitor cell that then, would differentiate, into kinds of, progenitor cells, and that would further undergo differentiation, right? So for instance, here we have T cells, the T lymphocyte, the B lymphocyte, and, natural killer cells occurring from the same progenitor, from the same ancestor right here. You could have dendritic cells, red blood cells, platelets, macrophages, granulocytes. Now, what has been recognized is that cancer cells kind of have a certain form of stemness in them. So just like normal stem cells, right, and stem cells, some cancer stem cells are able to renew, self-renew, and then differentiate into different kinds of cancer. And more awareness is kind of being thrown on this, because a lot of emphasis is being placed on cancer stem cells as the cause of the recurrence. So when there's treatment, so let's say a patient presents with a tumor or a cancer, there might be surgery to remove it if it's possible, and there may be radiation, and then they may go ahead with chemotherapy treatment to make sure they clear out any cancer cells. Cancer stem cells, however, seem to have a mechanism that allows them to stay. So under certain conditions, for instance, they can kind of their division rate So chemotherapy, essentially, most chemotherapies are targeting, cell cycle and then making sure that, it stops it and the cell will die. Certain cancer stem cells would actually, stop dividing. So they lower their threshold for division so that these chemotherapeutic agents are not able to act on them. And once, the treatment is gone, they then develop that kind of, sensory memory to overcome that chemotherapy by developing certain kind of, resistant, features, right? So this is a major aspect of resistance that we know of in cancer stem cells. And one major driver, genetic driver of cancer stem cells that I've been interested in is the gene called SOX2. So SOX2 occurs in normal embryonic stem cells, right? So they are very important for maintaining the stemness in normal cells at the very early stages of life. Similarly, SOX2 has been shown in certain cancer cells to be overexpressed. So unlike in embryonic cells where um, SOX2 expression is high early on in life, but then it's essentially, it's non-existent as, the person undergoes, development into the adult. In certain kind of adult tumors, you can actually have a lot of SOX2 occurring, right? So this essentially kind of, points us to possibly SOX2 playing a role in the stemness of the cancer stem cells. And it doesn't just do it alone. SOX2 usually cooperates with other genes. So for instance, here OCT4, NANOG, and SMAD1/3, for instance, are also a network of stemness factors that promote the development of the cancer stem cells. And over here, what you have is a set of genes that are associated with the potency, like the ability of the cancer cells to just, keep on growing and dividing. And the ones in red are the genes or the proteins that are whose expression are promoted by SOX2 signaling. And the ones in green are those that are inhibited. And as we can see here, SOX2 expression, so essentially this signaling affects all of these different targets and these targets that are affected also then, are affected or affect other signaling pathways. So the SOX2 signaling pathway essentially also affects, other signaling pathways, which is why it wouldn't be just sufficient to, let's say, target SOX2 because it's overexpressed. It's also important to understand why these other signaling pathways are connected to SOX2 so that in trying to target SOX2, we can also, target these other pathways and make sure that there are no compensatory mechanisms that promote, a recurrence when that happens. So this is one example of, how our systems biology approach, understanding of what happens with one gene, let's say overexpression, will then result in other genes being affected, overexpressed, or downregulated, which would in turn affect some other pathways, and how targeting these different pathways, either individually or collectively, may be much more effective than just doing, just targeting, let's say, one gene. All right. So in a study that was done by a collaborator of mine back in 2012, what they did was essentially to take tumor cells, and these were lung tumor cells, and different presentations of it. So there's a squamous cell carcinoma, this is a adenocarcinoma, and then they looked at different stages of the tumor. So stage 1 and 2 are lower stages. They are kind of much more less aggressive than stage 4, right? And what they see essentially by staining sections of the tumor cell, right, for this SOX2 protein, what they see is a lot of, SOX2 protein expression. So we can see, the brown coloring here is a SOX2, right? And then you can see that it's much more, a lot more, in the late stage cancer. And here, when you take the adenocarcinoma, it's much more pronounced than you have in the middle stage 1 and 2. And then when you look at the metastatic adenocarcinoma, for instance, here in the lymph node, you have in the bone, you have the adrenal, and then the brain. Again, you see expression of the SOX2. Now remember, as I said, so in the normal cell, embryonic cell, is supposed to just play a role in development and then essentially just kind of, lay low and just get out of existence. So for it to be existing or caring in cancers, and most of these, adult type cancers, in very large amounts is troubling, right? Which then tells us that it's playing an important role in these tumors. And so when they did the quantification, the positive staining, and you can see here the positive staining, you do have a whole lot, right, compared to the total number of cells around. The early stages, about 67%, and then you have the late stage 4, you have 73%, and then the metastasis compared to the control, you have a higher percentage of staining. So obviously SOX2 has a role to play, not just in the development of cancer, but in the aggressiveness of the cancer as well, right? Um, and this essentially is the graph comparing the the early stage and then the late stage, and then again looking at the primary side and then the metastatic side, and then you see a vast difference, very significant. Um, so this essentially is telling us that SOX2 is very important as a promoter of cancer. And in this study, in this graph, what the treatment that were given, these are 41 cells uh, that were treated with a scramble. So this is a controlled short hairpin RNA. So this is silencing, to silence the gene, but this has no effect. It's just a mashup of silences, and um, this is a specific SOX2 silencer, right? And in here, what they recognized was that a lot of tumor cells or tumors recruit macrophages, and these tumor-associated macrophages actually promote the aggressiveness of the tumor. So here, they included macrophages, right? Injected macrophages together with the treatment here, the control treatment. And here, they added the macrophages together with the SOX2. And in treating the macrophages, they use a pre-treatment with this drug, I'm sorry, this is essentially a pre-treatment with interleukin-4. So that essentially promotes the recruitment of the macrophages, right? And so what you can see over here, compared to the control, right? So the tumor size, the volume is high when you have recruitment of macrophages. So it's actually promoting a much more aggressive tumor. And then you have when it's treated with the SOX2 shRNA, right? In the presence of the recruited macrophages, you have a drop of the tumor size, so it comes down. But then when you have just no macrophages, just a tumor treated with the shRNA, you have a significant reduction of the tumor compared to the control. So right here, we can see that just doing a monotherapy, just targeting only the SOX2 with a specific molecular target, right, results in reduction of the tumor that was developed. And then here they did the staining to show, in normal cells, some markers of tumors, so CD163 and SOX2. And we can see here that in the tumor, there's, a high amount of the CD163, and then, high staining of SOX2. So again, underscoring the importance of SOX2 in promoting the cancer cells. And this is a study that we published in 2019. Again, we used SOX2. So what we did was to develop two models of lung cancer. In one model, we took mice and then we injected cancer stem cells. So these were lung H1650 cells that had a high expression of SOX2 in it, injected into the lung, so a primary site in the lung, right? And then the other one, this is a xenograft, where we took the same injection, but instead we injected it on the, subcutaneously, right? So on the dorsal side of the mice, so we could see it, right? Unlike in the orthotopic, where you would have to sacrifice the mice before you can see the tumor. And what we can see right here, so these treatments, the ABCD, there's the control, right? And there's the treatment with the cisplatin, which is a chemotherapeutic agent. It's a platinum compound that essentially disrupts, cell division, the cell cycle, and results in death. And we use a normal therapeutic, dose, that we would use in an adult. And then here we have our SOX2 sRNA. So this is a molecular target of SOX2. Here, it was actually a very small amount of cancer. We had a lot of skin attached. So it was kind of misleading. And similarly here, so you can see in this graph, when we quantify the total weight of the xenograft tumor, that there was a reduction, right, in the cisplatin, right? So there's like chemotherapeutic agent, there was a reduction. And there was a similar reduction in the SOX2 targeted group, right, compared to the control. And then when we combine the SOX2 and then the cisplatin at reduced concentration of the cisplatin, we had a much more enhanced effect. So this was significant compared to each of these groups and significant compared to the control. Now the question that someone would ask is, what's the use? I mean, if chemotherapy is essentially, promoting as much, tumor regression as a molecular targeting, why bother? And the answer to this was that one of the problems of chemotherapy is its lack of targetability. So it's not very specific. Most chemotherapeutic agents, just would diffuse into, all the cells. So they may end up in normal cells. And a lot of patients may actually end up dying, not from the cancer itself, but from the treatment, the chemotherapeutic treatment. And actually in this study, the treatment was done, over a course of 14 days. For the cisplatin group, by day 8, all the mice had died. In fact, they had lost so much weight, we had to sacrifice them. So we could see right here that it's just not sufficient to have chemotherapy if you have all these attendant, side effects. And when we had, a combined, targeted therapy as well as our chemotherapy at reduced chemotherapeutic dose, we had a much more enhanced effect. So this kind of, goes to support a much more dynamic and, combination approach to, treating cancer. Instead of just using one therapy, you could, combine different therapies and improve, the outcome, while also limiting the extent of resistance occurring. The tumor cells may be able to identify certain aspects of the one treatment and develop, capabilities to, evade them. But it's really difficult when you have two different treatments that have different mechanisms for, resistance to occur. Very well. We are almost there. So the other aspect, I just finished talking about the tumor resistance and the cancer stem cell paradigm, or the theory that underlies that tumor resistance, and how targeting, for instance, SOX2 signaling, right? For instance, targeting SOX2 was shown to result in regression of the tumor. Another aspect of tumor, which enables the growth of these tumor, is what we call dynamic rewiring, right? So as you can see here, this is a very elaborative, very elaborate, mechanism that identify different circuits, right? So different components of a tumor cell, right? Different genes are playing different roles, and some of these genes are involved in promoting metastasis. So these fall under the motility circuits, right? And then you could have other gene families or groups or signaling that essentially promote cell division, right? So that would fall under cytostasis or differentiation circuits. And then the viability circuits, right? That also promotes growth. And then you have proliferation, circuits, right? So all of these different, and there's just, a simple kind of, trust me, when you see an elaborate, signaling pathway, for instance, a SOX2, where you have different, markers involved, this is very, very simplified. But in this, you have all the six different, hallmark capabilities playing a role, right? Which will then result in all the changes that we see in the gene expression. For instance, in MYC, which is an oncogenic protein that is involved in very aggressive tumor and promotes, metastasis of a lot of tumors, right? And a whole set of other markers. So you have these circuits that work together, and sometimes they are very distinct from each other, but also connected to one another, such that when you try targeting one circuit, you may end up, even though you may be, successful in targeting and, blocking the circuit, other circuits may end up being, activated. And so again, that kind of, introduces the challenge of how do you target all of these aspects of achievement. And that's an ongoing, exercise that we're still engaged in. And so to kind of, emphasize the theory of, the concept of dynamic rewiring, for instance, we can look at, for instance, different treatments. So on the left, you have, different chemotherapeutic, treatments, plus, irradiation right here. And then here you have what we call targeted therapy. So these are treatments that target either specific signaling pathways. So for instance, erlotinib is a receptor tyrosine kinase that targets the EGFR signaling, right? Just as gefitinib. And what in treatment in the clinic, for instance, you can combine, different targeted therapies, or it's best to combine different targeted therapies with chemotherapy, just so you're able to target all the different circuits, right? Instead of just targeting one circuit. If you use erlotinib, you are targeting, the growth, static, circuit. But then if you introduce doxorubicin, you then, targeting the proliferative circuit, because it's, essentially prevents division and of the cells. So this is the concept essentially. And in this study, in this graph, for instance, to kind of emphasize how important it is to, one, combine these therapies when you have an understanding of the different components of the tumor cells and how those different components or circuits are working together. It's also important to understand that the effective treatment of cancer has to take into consideration, both the spatial and then the temporal, both the physical and then the time properties of the cancer. Now let me kind of explain it this way. So here you have single treatments here. At 8 hours, you have erlotinib, which is right here, was added, and then the number of cells that were underwent apoptosis were determined. And then at 36, you can see that number of apoptotic cells go up. Here's a single treatment with doxorubicin, which is our chemotherapy. And then here there's a combined treatment of chemotherapy and erlotinib concurrently. So they are simultaneously added to the cells, and then the number of cells that died or underwent apoptosis were then determined. But here, what the researchers did was to pre-treat the cells with the targeted therapy, with the erlotinib first, and then afterwards treat with the doxorubicin, right? And then what you can see here is that when you do the pre-treatment with erlotinib separately and then do the doxorubicin treatment later on, by 24 hours, you have a much more higher, almost more than 50% of the cells are dead. However, look right here. When you pre-treat with a chemotherapy agent, and then later on follow it up with erlotinib therapy, you don't see much of an effect occurring. So this kind of essentially underscores the temporal, the time component, right, of the treatment, different cancers may have different ways that some of the circuits may be playing a secondary role, compared to another circuit. So in this case, for instance, the reason why the pre-treatment of alertinib followed by doxorubicin was effective was because it was important to target the cytostatic circuit first. And once that had been targeted, then the proliferative circuit, right, was able to be targeted with the doxorubicin. When you did the reverse, by targeting the proliferative, there wasn't much effect. And so this again, this dynamic, it's almost when you see this data, you're like, oh my, what next? Because it's almost like every different types of cancers, we have very different intricate, properties that have to be individualized when you are treating them. And that might be much more effective ultimately in eradicating the tumor than just, deciding, hey, genes are overexpressed, or there's, overexpression or increased growth of this, cancer cell. Let me just combine these chemotherapy and targeted therapy, and that'll be it. Sometimes, the sequential treatment may be much more effective than concurrent or simultaneous treatment. And same here. So right here we can see number of apoptotic cells. You see the pre-treatment, and then you have that. In BT20 cell, this is a triple-negative breast cancer, very aggressive tumor. But then when you come to a different kind of tumor, so in this case, you have an expression. It's not triple negative. You have overexpression of HER2. When you do the same treatment, you actually don't see a replication of the results. So different, same breast cancer, cells, different, subtypes, and they have different results. And then here you have a luminal breast cancer, and you can see kind of this tracks very much with the TNBC, all right? And then when you take the normal cell, essentially, you are seeing essentially, no effect. So you have the cells, different subtypes responding differently, and it's important to identify what specific type of tumor you have, and then the specific targets that are, promoting that tumor in order to essentially come out with how to treat them. Right. So just kind of, so here again, in talking about how treatment of our cancer cells. So we take our BT, the TNBC cell, and then the HER2 overexpressed cell, and you can see how much, what different signaling pathways are affected, right? So you see the overexpression here, high overexpression, right? And then you see all the different pathways that are affected or that are being affected, or that are taking place in this tumor cell, right? So very many. And these signaling pathways kind of interact with each other. There's kind of crosstalk. So sometimes, you target the signaling pathway, and then the other signaling pathway, hears about this pathway being targeted, and they're like, hey, okay, we have to, essentially, cover our backs, right? In tree, there's a saying that, I don't know how many of you can speak tree, but if obi about share no what's I mean, loosely transliterated, if you see someone's beard burning, you get a bowl of water and put it under your beard, just so you can douse it when yours catches fire. So these kind of mechanisms present very different aspects of treating cancer. So it's not done. The work of the Cancer Genome Atlas, for instance, was supposed to help us identify all the genes that are implicated in a particular kind of, tumor. They sequence all the genome, but then even with those data, you still have to look at how these different genes are interacting with different other genes and other signaling pathways in order to be able to affect treatment. And similarly here, we see very similar, effects. So you have different proteins and pathways being affected differently across different breast cancer types. All right, I'm just, very slow. I'll just kind of think we spent enough time here. So here, I just wanted to share with you some data that Dr. Comensa was able to generate. As you may very well know, Dr. Comensa, is his area of scholarship is in computational biology. So he does a lot of genomic data analysis. And here, what he was interested, and we're working collaboratively, again, in trying to kind of integrate the therapy or the targeting and then the target component, right? We're interested in identifying the genetic differences between breast cancer occurring in Blacks or African American patients and white patients. And what had been shown was that, you know, Black or African American patients present with a much more aggressive kind of cancers, right? And when he was able to curate all those different genes or gene sets that were, implicated in these different cancers, what we see is that there's um, an abundant expression, right? An abundance of certain genes. So what you see in the red is more of these genes are overexpressed in the blacks compared to the white, and most of these genes are involved in promoting epithelial to mesenchymal transition, which is a hallmark of metastasis. So very aggressive tumors have high potential for EMT. All right. So in being able to identify these gene sets, these genes that are kind of implicated in this, we then are able to kind of and then try to figure out, is there a pathway, for instance, that they are involved in, and how do these pathways, how do these genes in the pathways interact with each other and with different pathways or signaling, right? And in this other study, what he did again was to identify again the genes that were overexpressed, and then we saw some oncogenes, for instance, TET. I made mention of TET earlier on. And what we then tried to do was to figure out how TET, essentially promotes cancer, right? So we see that an overexpression of TET was associated with an increased expression of c-Myc, and c-Myc is, component, it's an oncogene, again, involved in the promotion of aggressive metastatic cancer. And which also promotes the overexpression of a certain microRNA, right, that affects the expression of a particular gene that is involved in cell cycle, right? And so you have all of these different components or these different genes that are interacting or that are associated with TET. And in trying to target TET and hoping that be able to block these pathways, it's also possible that another approach may be to target some of these other pathways, right? And so again, just trying to emphasize how dynamic the whole concept of our targeting is. I'm almost done. So here, this was a study that a student of mine, a master's student, Banda, did. He's currently doing his PhD at Wayne State University in Michigan, where we were trying to look at lung cancer cells and essentially, the aggressive form of lung cancer and how effective, targeted therapy. In this case, we have erlotinib and gefitinib, which again are receptor tyrosine kinase inhibitors that target the EGFR pathway. And we can see that a very large amount of drug, the IC50 is quite high, is needed, in H1975 type of lung cancer cells. And when you come to H1299, a much more, larger, amount or dose. So this is very, very, H1299 is very, very resistant. And here, what he did was to take a chemotherapeutic agent, computacin, and then try to look at, combination of the targeted therapy and low dose computacin, how that might or affect treatment. So we can see the IC50 of camptothecin is 7 micromolar. When we used a low dose camptothecin of 0.5 micromolar with erlotinib, we ended up drastically reducing the dose of erlotinib that would cause the killing of 50% of the cell. So here again, a certain form of combination therapy, but it also underlines the dynamic rewiring or targeting of the different circuits, right? And here, we looked at different targets that we wanted to see. For instance, salinomycin is a chemical compound that targets SOX2. And then this compound, ALWII4127, targets another receptor tyrosine kinase ephrin A2, right? And then we have a PD98059, which is an inhibitor of PI3K, and LY294002, which is an inhibitor of the ERK-MEK pathway. So different signaling pathways are being investigated right here. And the reason why we looked at that was because based on literature, we found out that the EGFR oncogene, for instance, promotes the overexpression or transcriptional regulation of this F2, and the EGFR had also to promote expression of SOX2, which in turn also promotes the expression of FA2, thereby introducing a positive feedback loop, right? So what we were interested in finding out was, is there any connection between our FA2 and then our SOX2, right? So we have three signaling pathways and try to find out how these work together. The reason why we're interested in FA2 was because we recognize playing, roles in promoting migration inflation and then cell proliferation, all of which promote the cancer growth. And briefly to put it together, this is what we found out. We saw that the PI3K pathway was affected, or the SOX2 gene or protein was affected by treatment with our FA2, which is the ephrin A2 receptor target, and then treatment with a SOX2, as well as treatment with a AKT pathway inhibitor, right? So these three different signaling pathways affect SOX2 expression, and then we see similar, occurrences happening. So the point that I want to make over here is that within just the treatment in trying to target SOX2, for instance, you could actually trigger other pathways because of the feedback loops that may be occurring. And so it's much more important to have a much kind of holistic way of targeting it, where you may have three different treatment targets: your SOX2, your FA2, your EGFR, so that you kind of blocking all the different circuits that are promoting the So I'm going to end here, and then I want to thank you for your time. I think I've taken quite a bit of time, and would like to open the floor for any questions. But before then, there was a question that was sent earlier. And the question was, how do we eradicate cancer stem cells as they are reported to be resistant to chemotherapy and radiation? Since in theory, a single residual cancer stem cell can see the growth and metastasis of new tumors. So this is a very valid question, very important one too. If you have one single, cancer stem cell, it has a potential of, expanding into a colony, then, differentiating into different kinds of tumors. And so it's important not just to remove the, large mass of tumor, but also to remove this residual, cancer stem cells. And one of the ways would be to be able to specifically target the marker that is promoting that stemness. So for instance, based on, for instance, sequencing of the tumor, and then identifying that SOX2, for instance, is overexpressed on mutation, we can develop a specific target, such as an shRNA or target that specific SOX2 or OCT4 or nanog or whichever, stemness factors it is, and then make sure that that residual cancer is eliminated. The second question is, how can we distort the normal cells of the body from nourishing tumor cells so as to starve them to death? So this is very important. Now, the second slide that I showed you that had the, conceptual image of a tumor cell, you remember I showed you that, and then it had those blood supplies coming out. Very early on, our understanding of angiogenesis was that cancer cells would just co-opt, will kind of, hijack the normal blood supply of the body, and then, use it to nourish itself. Later on, we kind of, became aware that the cancer cells actually have the capability of developing their own, blood supply. So this is a very big challenge. It's a very big challenge, but there are therapies, right? And these are essentially what we call the anti-angiogenic therapies, which target the factors or the markers that promote the, formation of new blood cells. So these may be, for instance, VEGF, vascular endothelial growth factor is an important component of, development of new blood vessels. And there are therapies that target, VEGF, for instance, specific targets that would then, eliminate those. For instance, afatinib, some receptor tyrosine kinase inhibitors would target those. And so, our anti-angiogenic treatment would be able to help in starving the tumor cells from growing and developing. The third question that I had was, what key concepts in math and biology are required to have a deep understanding of the systems approach to developing the formulation? Generally, from the work of the Cancer Genome Atlas, and if you go on the website or the website of any of their consortium partners, you would see that they have, tutorials. So yeah, it's great to have, basic understanding of some of the concepts, that you need in math or biology or physics or, so many different engineering to be able to do it. But essentially, just basic understanding of in biology, right? Your system biology, in understanding the targets that you are interested in, is sufficient to start, because most of these programs have tutorials that will take you through the different steps. And Dr. Akomensa has had many opportunities of training our PharmD, students who are actually in the full-time PharmD program, and decide to work with them, and within a term, a semester, they've gained some understanding of genomic analysis and how to interpret data and all that stuff. So it's possible to do with some very basic understanding of your biology, your oncology, and then obviously some computer skills. And then once you're able to follow those tutorials, you'll be able to do that. I'll now open the floor for anyone who has any question.
Thank you very much. Thank you very much, Professor Andey. This has been a very enlightening presentation. We'll open the floor for questions. So there's a place where you can raise your hand. If you click on raise your hand. All right, while they contemplate questions, let me just ask this one. How developed is the RNA interference technology, siRNA, and to what extent is it available in the clinic?
Oh yeah, it's very much available. So most of these, because these are almost like personalized treatments, in terms of manufacturing and the cost benefits, it's difficult to kind of make on the shelf preparations. So mostly formulation scientists would come up with the drug formulation, let's say a liposome, in the figure that I showed about the work that we did. For instance, we encapsulated the SOX2 siRNA in a liposome. So you could actually make the liposome as a delivery system maybe lyophilized and have it in a vial. And then if a patient presents with a tumor and then there's a tumor resection and then there's the analysis of the tumor sample and they recognize certain kind of genes that may be promoting it and they would want to target that, they could then design the siRNA from a gene library for the target. And once we have the siRNA, we can then, for instance, dissolve it in an aqueous mixture and then introduce it into the liposome, get it encapsulated, and then inject it into the patient. So right now we don't have a whole lot of on-the-shelf products, but in the clinic, especially in these cancer centers, they have these technique or application where they can just design right there, design a therapy, molecular therapy, and then put it in an appropriate dosage form and then and present it to the patient. And it's very similar to the CAR T cell therapy, which is actually approved, chimeric, treatment for cancer, where they take the patient. So essentially, they'll take the patient's, T lymphocytes, blood samples, and then isolate lymphocytes, and then change, the lymphocytes in order to be able to target that specific tumor, generate the antibodies, and then they will just, infuse it back into the patient. That's like where we are right now clinically.
Terrific. Thank you very much, Dr. Andey. I have a question from Isaac. Isaac from KNUST. So in the earlier part of your presentation, you mentioned that cancer cells have the capacity to create their own blood vessels. How can targeting therapy as preferred over chemotherapy be able to curb such a situation?
So I for a long time we kind of gave chemotherapy a very bad name because we were always talking about the side effects and how toxic it was and all that stuff. But what we've realized is that we really need chemotherapy, as bad as it is. And I think the balance that has to be struck is optimizing the dose of the chemotherapy and improving how selective and specific it is. And this is where the formulation comes in. For instance, we have very elaborate formulation techniques where we can make the formulation, let's say liposome, and then have a target, let's say a protein that will bind to a specific receptor or overexpress on the tumor. So that introduces a certain, kind of selectivity to the process where you can improve, the selective nature of your treatment, and then reduce the dose, right? And then you can then combine that treatment, that chemotherapy treatment, with a molecular treatment, right, or molecular therapy, such as your anti-angiogenic therapy, right? And so we can, again, based on understanding of the dynamic rewiring, right? Which comes first? Is it the egg or the chicken? Is targeting the blood supply more important than targeting the tumor? And when you rationalize it, you realize that when you're using chemotherapy, you need it to get into the tumor first. And so my guess would be that I'm going to use the chemotherapy, get it into the tumor, and then I'll use the anti-angiogenic agent to then block the formation or recruitment of my VEGF, so that once the tumor cell has taken up the chemotherapy and is being destroyed, I'm blocking the supply of the blood with my anti-angiogenic therapy, and then I'm effecting a much more enhanced outcome. And so think of it in terms of a more cooperative approach, more combination approach, instead of different distinct approaches. I hope that answers the question.
Yes, it does. Are there any other questions? Okay. In the absence of further questions, we'd like to thank Professor Andey for this wonderful presentation. It's recorded, and we'll decide later on what we do with the recording. But we thank you all for your participation. We'll later on send a feedback form so we can get some feedback as to what kinds of topics you'd be interested in and what would be next. All right. Thank you all very much. And thanks very much, Professor Andey. Thanks.